Hi, welcome to Power Medicine. Today we're going to be discussing with respect to glycolysis, the first pathway of carbohydrates metabolism. First of all, let's have an overview on it. The term glycolysis is derived from two words. Glyco means sugar or glucose, and lysis means splitting or breaking down. And the following key points about the glycolysis. This is the first and best understood metabolic pathway, comparing to the citric acid cycle and other pathways. This is an enzyme catalyzed pathway. It means all its reactions take place inside a cytosol in the presence of different types of enzymes. All the enzymes catalyzing these reactions are found just inside the cytosol of the cell, whereas the Krebs cycle and electron transport chain enzymes are found inside the mitochondria of the cell. Glycolysis consists of 10 successive reactions from 1 to 10, giving off different types of intermediates in this process. These 10 successive reactions of glycolysis is then divided into two phases, which is preparatory phase and payoff phase. The preparatory phase is also called investment phase. In the process of glycolysis, the molecule glucose, which is a 6-carbon molecule, is converted to 2 molecules of pyruvate, which are 3-carbon molecules. And all these reactions take place just inside a cytosol of the cell, whereas the citric acid cycle and electron transport chain take place inside a mitochondria of the cell. During the glycolysis, a fraction of energy is released in the form of ATP and NADH, which is the reduced form of NAD. And the best thing about the glycolysis is that it can function both aerobically and anaerobically, and there is no severe necessity for the presence of oxygen. First, let's have a quick glance on the intermediates that are generated during the two phases of glycolysis. And we will discuss every single reactions more in details along with their specifications later on in this video. In the first reaction of glycolysis, the intermediate generated is glucose 6-phosphate, which is the first phosphorylated intermediate in this series. Then it can be isomerized to fructose 6-phosphate, a phosphorylated ketose which is generated within the second reaction of glycolysis. In the third reaction of this series, fructose 1,6-bisphosphate is generated, which is then broken down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, both having a phosphate group representing reaction number 4 of this series. In the fifth reaction of glycolysis, the hydroxyacetone phosphate is isomerized to make a second molecule of glyceraldehyde 3-phosphate out of 1 molecule of glucose. The sum of these first 5 reactions represent the first phase of glycolysis, which is called preparatory phase, or you can say investment phase of glycolysis, in which energy is just invested. The compound generated in the sixth reaction of this series is 1,3-bisphosphoglycerate, which is achieved from glyceraldehyde 3-phosphate. And this reaction is the beginning of the second phase of glycolysis. In the seventh reaction, 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate, and this is the first reaction of glycolysis in which energy is released in the form of ATP. The eighth reaction reveals generating of 2-phosphoglycerate from 3-phosphoglycerate. And all these reactions are basically catalyzed by specific enzymes. In the process of ninth reaction, 2-phosphoglycerate is dehydrated to make phosphoenol pyruvate by removing of 1 molecule of water from 2-phosphoglycerate. Finally, in the last reaction of this series, pyruvate is generated from phosphoenol pyruvate, giving off energy in the form of ATP as well. Likewise, the sum of these 5 reactions is called payoff phase, during which energy is reaped in the form of ATP and NADH. So ATP is achieved in the 7th and 10th reaction of this series. Now, let's have a closer look on every single reaction taking place during the preparatory phase of glycolysis to earn a better extra knowledge in this regard. When glucose molecules are absorbed from diet, they are stored in the liver and then propelled towards the cell for consuming. First, glucose molecules are transmitted inside the cell through a number of carriers in the cell membrane, and these glucose molecules are transmitted for metabolic purposes and as a fuel for the sake of energy production. Once they're transmitted to the interior of the cell and remain without of any modifications, they have the ability to go to the reverse direction as they came before, through the same carriers. And that's why they must be phosphorylated to become fully polar so they can't get out of the house easily. This phosphorylation is the first step of glycolysis. Step 1 is the first step in which glucose is phosphorylated. Glucose molecule is converted to glucose 6-phosphate, which is a phosphorylated compound and fully polar. The phosphate group attached to glucose 6-phosphate is achieved from ATP, converting it to ADP. This reaction is irreversible, and the reason why is that energy is spent here. The enzyme catalyzing this reaction is exokinase, more specifically you can say glucokinase. This enzyme necessitates magnesium ion for its activity. Structurally explaining, you can see here glucose structure which is going to be converted to glucose 6-phosphate by attaching of one phosphate group from ATP to carbon number 6 of glucose. Again, this reaction is catalyzed by exokinase. Now, what about an exokinase? Exo means 6, and kinase has to do something with transferring a phosphoryl group. And here, phosphate groups is transmitted from ATP to glucose. In the second step of the glycolysis, there is an isomerization occurs to glucose 6-phosphate, finally makes it fructose 6-phosphate. This is an active group isomerization, converting an aldose to a ketose. This reaction is reversible and can go to the heater sites, because for the next reactions we need fructose 6-phosphate. This reaction goes to the right side. This reaction is carried out by the presence of phosphoexose isomerase. Once again, the presence of magnesium ion is considered in this reaction. Looking more in details in the structure of glucose 6-phosphate, having a phosphate group on carbon number 6, which is supposed to be converted to fructose 6-phosphate, is still a phosphorylated compound, but is a ketose, not an aldose anymore. And of course, this is a reversible reaction catalyzed by phosphoexose isomerase. Exose stands for 6 carbon compounds, which represents sugar here, and you can also say phosphoglucose isomerase. Here we are in step 3 of the glycolysis. We have fructose 6-phosphate that has been isomerized from glucose 6-phosphate, and in this reaction, this is again phosphorylated to make fructose 1,6-bisphosphate, having 2 phosphate groups instead of one phosphate group on carbon number 6 and the other on carbon number 1. The newly phosphate group attached to this compound comes from ATP, which makes it ADP and gives a phosphate group to fructose 6-phosphate. And this reaction is catalyzed by enzyme phosphofructokinase 1, abbreviated as PFK1, that needs for magnesium ion as its cofactor in this reaction. And now we're going to look at the structure of these intermediates to understand more. Here we got fructose 6-phosphate, which has a phosphate group on carbon number 6 and is phosphorylated again to fructose 1,6-bisphosphate, which takes this new phosphate group from ATP inside the cell. And this newly compound has 2 phosphate group, one on carbon number 1 and the other on carbon number 6. If 2 phosphate groups are attached together side by side, we use the prefix di, like in ADP. But if they are far away from each other, we use the prefix bis, like in fructose 1,6-bisphosphate. Step 4 of the glycolysis is the step in which breaking down happens. We got fructose 1,6-bisphosphate, a compound containing 2 phosphate groups, which is broken down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, both having a phosphate group attached to their structure. This reaction is reversible and mainly goes to the right, and fructose bisphosphotaldolase catalyzes this reaction. Here is the structure of fructose 1,6-bisphosphate with its phosphate groups on carbon number 1 and 6 that is going to be undergoing cleavage and produces 2 smaller phosphorylated compounds. And these 2 smaller compounds are glyceraldehyde 3-phosphate and hydroxyacetone phosphate that you can see their relevant structures here. Again, this reaction is reversible and is facilitated by the enzyme fructose bisphosphoraldolase. Actually, in this reaction, the lysis part of glycolysis takes place, and a cleavage occurs between carbon number 3 and carbon number 4 of fructose 1,6-bisphosphate, converting a 6-carbon ketose to 2 3-carbon molecules, an aldolase, which is glyceraldehyde 3-phosphate, and a ketose, which is dihydroxyacetone phosphate. Step 5 is an isomerization between products of the previous reaction. In this reaction, dihydroxyacetone phosphate undergoes isomerization to yield a second glyceraldehyde 3-phosphate to participate in future steps of the glycolysis. And this reversible isomerization reaction accomplished by the enzyme triose phosphate isomerase. Here you see the structure of dihydroxyacetone phosphate having a keto group which is isomerized to make glyceraldehyde 3-phosphate which contains aldehyde group in its structure. This is a reversible reaction isomerized by triose phosphate isomerase and mostly goes to the right side to produce glyceraldehyde 3-phosphate, and because the next reaction of glycolysis do not need dihydroxyacetone phosphate, it must be isomerized to glyceraldehyde 3-phosphate for further progress. And now keep it in mind that up to here one molecule of glucose produces 2 molecules of glyceraldehyde 3-phosphate, one in step 4 and the other here in step 5. Step 6 is the beginning of the payoff phase of glycolysis, a phase during which energy is going to be reaped rather than investing. In this reaction, glyceraldehyde 3-phosphate that has been produced in previous reactions reacts with one inorganic phosphate group, not an ADP that might be mistaken, to make 1,3-bisphosphoglycerate, a compound with 2 phosphate groups. This reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, and catalyze is a reversible redox reaction in which coenzyme NAD or nicotinamide adenine dinucleotide is reduced to NADH and receives hydrogen ions. If we take the structures into consideration, we can see here glyceraldehyde 3-phosphate which has a phosphate group on carbon number 3 and reacts with inorganic phosphate to make 1,3-bisphosphoglycerate by attaching this inorganic phosphate group on carbon number 1, facilitated by glyceraldehyde 3-phosphate dehydrogenase, also accompanied with reduction of NAD to NADH. The NADH generated here then goes to electron transport chain through different ways like glycerol phosphate shuttle or malate aspartate shuttle to produce more ATP, a process called oxidative phosphorylation. And remember that up to here we produce 2 NADH because you know that in glycolysis 1 glucose gives 2 glyceraldehyde 3-phosphate and 1 NADH is produced per 1 glyceraldehyde 3-phosphate. Step 7 of the glycolysis is very important that 1,3-bisphosphoglycerate reacts with 1 molecule of adenosine diphosphate inside the cell to produce 3-phosphoglycerate and the first ATP of this series. This reaction is catalyzed by phosphoglycerate kinase, and again kinase enzymes are related to the replacement or transferring of phosphate group, which in here phosphate group is taken from 1,3-bisphosphoglycerate and attached to ADP to make it ATP. Observing the structure, you can see here 1,3-bisphosphoglycerate that has 2 phosphate groups attached to it, and reacts with 1 molecule of adenosine diphosphate to lose one of its phosphate groups and makes 2 phosphoglycerate plus 1 molecule of adenosine triphosphate, which is catalyzed by the phosphoglycerate kinase. And again, keep that in mind that up to here 2 ADPs are produced, because you remember that 1 glucose divides into 2 glyceraldehyde 3-phosphate in the process of glycolysis. And up to this level, 2 ADPs are produced per each molecule of glucose. In step 8 of the glycolysis, 3-phosphoglycerate, which was produced in previous reactions, undergoes a little modification to be converted into 2-phosphoglycerate. This is a reversible reaction that is facilitated by phosphoglycerate mutase that necessitates magnesium ion for its activity. So let's illustrate all these in details here. You can see the structure of 2-phosphoglycerate that has a phosphate group on carbon number 3 and is converted to 2-phosphoglycerate that has got a phosphate group on carbon number 2. Basically, the process of this reaction is an exchange of phosphoryl groups within 3-phosphoglycerate and an active site of enzyme mutase. This active site of enzyme gives a phosphate group to 3-phosphoglycerate, makes it 2,3-bis-phosphoglycerate, and after that recaptures its phosphate group from carbon-3, which finally makes it 2-phosphoglycerate. In step 9 of the glycolysis, during a dehydration reaction, the 2-phosphoglycerate is modified to be converted into phosphoenol pyruvate. This reversible reaction is along with removing of one molecule of water from 2-phosphoglycerate and is catalyzed by the enzyme enolase. Looking to the structures, you see here 2-phosphoglycerate, which is converted into phosphoenol pyruvate by releasing of one molecule of water. This releasing of H2O or water molecule facilitates providing of ADP for the next reaction that phosphoenol pyruvate undergoes and produces ATP out of ADP. And finally, we get to the final step of this series. In this step, phosphoenol pyruvate reacts with another ADP molecule of the cell to produce pyruvate and another ATP. This reaction is catalyzed by pyruvate kinase that transfers the phosphate group from phosphoenol pyruvate to ADP. This enzyme also needs magnesium and potassium ions for its activity. Here we have got the structure of phosphoenol pyruvate that contains the phosphate groups attached to it. In a reaction with adenosine diphosphate that is found inside the cell, it makes pyruvate along with adenosine triphosphate as released energy. This reaction is catalyzed by pyruvate kinase that transfers the phosphate group from phosphoenol pyruvate to ADP. Actually, in glycolysis, 2 ATPs are produced in this level, because 2 phosphoenol pyruvate are generated out of one molecule of glucose, and every single phosphoenol pyruvate gives one molecule of ATP. And the pyruvate here can be converted to acetyl-CoA to go to the Krebs cycle for further energy production. Now, let's summarize the products of the glycolysis. During the 10 successive steps of the glycolysis, one molecule of glucose reacts with 2 molecules of ATPs, one in step 1, the other in step 3. 4 molecules of ADP, 2 ADPs in step 7, 2 other ADPs in step 10, and 2 inorganic phosphate in step 6. 2 in HDH, also in step 6. Which give 2 molecules of pyruvate in step 10, 4 ATPs, 2 ADPs in step 7, and 2 ADPs in step 10. 2 ADPs, one in step 1, the other in step 3. 2 in HDH in step 6. And also 2 hydrogen ions, also in step 6. 2 molecules of water in step 9. Now the net gain during the whole process of glycolysis are 2 pyruvates, 2 ATPs, 2 NADH, 2 water molecules, and 2 hydrogen ions. Alright guys, thank you for watching. If you like this video, don't forget to subscribe our channel for more videos.